hosting Dr. William Mair today, who's a professor at Harvard School of Public Health. And so he did his bachelor's and PhD at University College London in the UK. And there, during his PhD, he studied with Dr. Linda Partridge and worked on Drosophila, where he found that you can start dietary restriction at any age in the flies, and then they'll still extend the lifespan. So it's something that's very flexible. And then he did his postdoc at the prestigious Salk Institute for Biological Studies with Dr. Andrew Dillon, and there he worked on C. elegans and worked on mechanisms like AMP kinase and CRED. And so, as a professor at Harvard, he synthesizes PhD and postdoc work, and yet continues to innovate on new mechanisms of aging, where he's added mitochondria and fatty acids to his research directions. But most importantly, he trained my good friend Carlos Silva Garcia, who was a postdoc at his lab, got K99 grants, published well, and basically got job offers left and right. But in the end, he chose to become a professor at Brown University. So I'm quite thankful for that. And so please help me in welcoming Dr. Mair for his talk, Metabolic Flexibility and Healthy Aging.
Thank you. That was a great introduction. You should have written my tenure packet. I only just managed to stumble over the line of tenure. So actually, I'm going to talk a little bit about the fly data. So it's a real pleasure to be here. Does that sound too loud, or is that okay? Yeah. So I'm going to talk to you about my research size. There's a lot of diversity in the research in the audience. So there's not a lot of genetics and genomics, although my training is in genetics. My lab has become a kind of cross-section of different disciplines. There's actually quite a lot of cell biology today. But I'm going to talk to you about some of my stuff. And actually, you're welcome to interrupt me at any time, ask me any questions. And what I wanted to do was mainly focus on unpublished work from a postdoc in my lab who's also looking for positions. So hopefully this will be an opportunity for him to get some exposure. So this is I often start my talks with this sort of graph. So in the last century, we were phenomenally successful at adding life expectancy to humans, right? These are data just in the US here. So in the average life expectancy in 1900 versus 2000, and we added about 27 to 30 years of life expectancy, not in all demographics, but on average in the US, and this is true in most other countries in the world, which is amazing, right? And so all of this was done through public health. None of this was done by modulating how our bodies age, the rate at which we age. It was all done by getting rid of things that used to kill us in our youth, whether it's from simple things like clean water, education, vaccination, antibiotics, all these sort of things. And all that's really done is shift the distribution of when we die to much later time points. This really enabled many, many more of us to live into old age, and this is going up and up. We had a bit of a dip in the last 4 years, but we're back on track. So by the point of 2050, we're going to have 1.5 billion people worldwide over the age of 65, which is a phenomenal success. But really, that's also come at a cost, right? You can make an argument, and I probably shouldn't because I work at a school of public health, that public health has given you Alzheimer's disease. Because by enabling us to live into these advanced ages, which we never really experienced in the course of human evolution, we've got past the point at which our bodies have evolved capacity to deal with damage, to deal with stress. And so what the result of that has been is that this huge increase in the number of non-communicable diseases on the right here, which age is a risk factor for all of them. So by putting the numbers game just mathematically to many more of us surviving past the age of 65, well, that's a wonderful thing. Those added years we've given ourselves have not been particularly healthy ones. We've exposed this sort of built-in obsolescence in our systems and enabled a massive increase in these diseases on the right here. So really what the goal has to be now in this next phase of public health, I would argue, is to make those added years that we've added healthier ones, and this is a new kind of pandemic for public health. But how do we go about doing that? So the problem is the way we got rid of those diseases in our youth is not really going to work necessarily for these diseases in old age, because the way you experience disease in old age is different from the way you do when you're young. So this is a graph here just looking at the number of comorbidities of chronic conditions in different age brackets in people, again in the US. And you can see the older you get, you begin to have 1, 2, 3, 4 different comorbidities at the same time, all of which age is a risk factor for the but they have very different etiologies, right? So mathematically, even if you were to get rid of one of those diseases completely cure it, which would be a great thing to do, the end result is not a massive number of the increase of people who are completely disease-free, you just have one less chronic condition. So when we think about the way we want to try and move forward in biomedical sciences to try and think about prevention or treatment for these age-related conditions, we need to really think slightly differently about how we kind of target those conditions. To begin to think about age as a risk factor itself. And I work at a school of public health whose goal is to identify public health risk factors and modulate them, and see if you can think about modulating the extent to which age is a risk factor for all of these different conditions. So this is central to the kind of way that I think, and many other people maybe in this room and in my field think about, is that is age itself a risk factor that we can mitigate? Is there something we can do to vary the extent to which age is a risk factor for different chronic conditions? And so that sounds a bit like sci-fi, except for we know there are lots of examples in nature and in humans of exceptional ages, right? We know that there are centenarians in human populations who live to 100, obviously, and that might be due to a combination of luck, genetics, environment, behavior, many different things contributing to that. But they don't just live to 100 and have a drawn-out period of comorbidity in their old age. They actually have a really compressed period of morbidity. So they're really functional and independent right into their 90s and then fail quite quickly. Across biology, there's extreme examples of amazing ages, whether it's whales that live 250 years, these clams that can live 500 years. You all know about naked mole rats, all these sort of things. There's lots of examples in biology of systems and organisms which deal a lot better with aging than we do. So can we learn about that from looking at exceptional ages in people or in different organisms and trying to understand why it is that some of us are doing less well? Kind of top-down approach. And then the other approach, which is really what my lab does, is take something which ages really poorly. So in this case, it's a nematode worm, C. elegans, that lives and ages and dies in about 3 weeks, as you all know. And then look if we can extend the period of longevity in those animals, extend the period over which they're kind of healthy, if you can define health in those worms. Look for conserved mechanisms that might also work in higher organisms, move through the ranks of Drosophila and mice. And then ultimately see if you can learn something about human aging by taking this bottom-up approach. And these two top-down and bottom-up approaches are not mutually exclusive, they're really complementary, and you can do this now really well. And so this is kind of now how we've coalesced around this word, which I don't love, but we're stuck with it now, geroscience. And that's really what I think the definition of geroscience is. So unlike gerontology, which is trying to understand just diseases that arise with old age and treat them, geroscience is trying to think more fundamentally outside of individual diseases and think about aging as a risk factor for multiple chronic conditions that you can mitigate, and try to think about therapeutic, behavioral, pharmacological, whatever they would be, policy-driven changes which modulate the extent to which age is a risk factor for all those diseases. And so this concept, it only works, right? That's basically saying it's better to cure all diseases than one, which sounds a bit sci-fi. It only works if aging and the links between age and disease and healthiness is in fact malleable. And for a long time, people kind of would laugh at that concept, even though we all know, we can all think of examples of people that we know that are aging better than others, some families that age better than others. You can see diversity in the rate of biological aging right in front of you with your friends. Certainly when I get to my 40s, I can now see my friends are aging better than others just physically. Right? We also know that the things that your grandmother told you to do, eat well, exercise, will really change the way your body ages. So we know that intrinsically that aging is malleable. But really what's happened in the last 20 years around the field of geroscience is we can show in the lab that in model systems, we also have many ways we can modulate the rate of aging and the links between aging diseases in the lab, whether it's through genetics, drugs, or diet. And the reason I say diet is really this idea of dietary restriction, which is a very broad term, which can encompass many, many things. I'm happy to talk about that at the end. But basically, its essence is some form of reduction in nutrients without malnutrition from ad libitum. Some form of dietary restriction has been shown to slow aging, increase the lifespan, and when you, in all of these organisms that I've put here, including the obvious genetic model systems, yeast, flies, mice, worms, and mice, but also we have longitudinal studies in non-human primates showing that dietary restriction can slow the rate of aging and then protect against those age-related conditions. So dietary restriction can really delay the onset or reduce the severity of these different age-related conditions. And in all these instances, this is my dog Scout here, so I don't do dietary restriction on her, but you can show the dietary restriction in Labradors. Also, she's actually a mutt, but will slow aging. So in all of these things, apart from us, we know there's a really strong way to modulate and affect the rate of aging through diet without having to do anything to genetics. And so this begs the question, is this a therapeutic intervention? And there's lots of public interest now and private donors interested in ways you might modulate diet, whether it's time-restricted feeding, intermittent fasting, these sort of things in aging. And I'm not going to talk about those today. I would argue that they are not a therapeutic intervention, and there's very little data for that. But there's also many side effects in a lab of a diet-restricted animal. It might live a long time. It usually has many different physiological costs associated with that. So what I've been interested in the last really 20-something horrible now, number of years, 25 years or 20 years anyway, is to understand the molecular mechanisms underpinning the effects of diet restriction on aging, to understand if we can tweak some of those, target some of those directly to get the beneficial effects on these changes to diet without actually having to change the diet itself. And as was alluded to in introduction, and I've now put these data in for reasons I haven't published and I haven't presented this work. This is 20 years old. This is the first ever paper I ever did as a graduate student. So if you're a graduate student, keep going, because one day you'll get so exhausted you'll hate presenting your data, but then 20 years later you'll come back to it and you'll like it again. But my grad advisor is recently retiring, and so I had to go to her retirement symposium and give a talk. So I just wanted to go back to some of the data. And so I've kept it in my slide because it really did, for me, kind of birth my interest in these nutrient and energy sensors which switch animals between different longevity states. So these are some data in a fruit fly from, my goodness, 20 years ago. This is age-specific mortality on the y-axis. So this goes up with time. It's basically the chance of dying at any time point in an animal's life or human's life. It's the sort of statistic we do to understand the increased risk of death of, say, something like smoking. It can go up, it can go down when you change that behavior. So this is a statistic that can go up and down as an animal ages. And if you let fruit flies eat as much as you want, and these are in chambers of 100 or 150 animals, so quite dense conditions, the chance of dying goes up with time, and then they're all dead by about day 35 in these conditions. If you diet restrict those animals, they still actually get complete access to the food, but you reduce the kind of concentration of all the nutrients in the food. You can reduce that risk of death. They're less likely to die for a long period of their life. Then eventually they will begin, that rate will go up, and they'll die as much as they want to increase their lifespan. But what I did in this study was move them between these different diets at different ages, as was alluded to in the introduction, and ask what happens. Does eating a lot of food cause some permanent damage, some, with molecular changes that you can't reverse and that you can't do? So even if you apply diet restriction late in life, maybe you just slow the rate of damage from that point on, or can you actually rejuvenate those animals? And what was kind of wild in fruit flies, if you applied DR at different times in their life, early age or middle life here, the equivalent of, I guess, me, within 2 days, these animals completely switched mortality trajectory. So that within 48 hours, you couldn't tell the difference between a fruit fly that had eaten ad libitum all the way up until 2 days ago and just been diet restricting itself for 2 days, from some that have been on DR all the way through life. Sadly, the converse is also true. If you went from DR to ad lib, you lost all the benefit very quickly for a fruit fly, and it's definitely not clear, and it seems maybe unlikely that this sort of rapid switching in mortality trajectory is going to be true in mammalian systems, certainly in mice and humans at late ages. But what this really meant is that there were built-in kind of switches in these animals that you can trigger by changing their diet, which completely alters their interaction with time and their experience of their aging trajectory, and can put them on a whole different path. And for me, that was very compelling, and I spent really the most of my career past that point trying to understand what these switches are, how we might target them, how they acutely might be thrown in different times of life, and how we can modulate them for aging interventions. And my lab now, which is, I started my lab in 2011, so kind of 12 years or so, has focused a lot on these two central energy and nutrient sensing kinases. So this talk's going to be a bit of biochemistry, not much really, a bit of genetics, a lot of cell biology. But we're really interested in these two kind of reciprocal antagonistic kinases. So on the left here you have AMPK, which senses low energy. So if you skip breakfast or you went for a run this morning, your AMPK is very active in all of your cells, it's conserved in all eukaryotes really. So it's activated by fasting, activated by diet restriction, and it promotes catabolism. Its job really is to turn off things that use energy and turn on things that generate energy. It's also pro-longevity. So if you take a variant of AMPK which is consistently active and you express it in a C. elegans or a fruit fly, or now also killifish, not I think in mice yet. That's efficient to mimic the effect of dietary restriction without changing the food intake. And on the flip side, you've got this kind of feeding activated kinase mTORC1, which senses high nutrients. It senses many things upstream of it. So it senses amino acids, growth signaling, oxygen, many different things. It's therefore turned on in many cancers. It's turned on under feeding, and it's suppressed therefore by dietary restriction. It's anabolic, so its main job is to regulate growth and drive all the building blocks for growth in the cell. And it's pro-aging, so if you inactivate TORC1 through genetics, through drugs, in multiple different organisms, that's enough to mimic the effect of dietary restriction and modulate aging and slow aging of species. So we've been really interested in understanding the interplay between these two, both at a kind of mechanistic level and where they work in an animal to really modulate aging, and how much they are mitigating some of the effects of dietary restriction longevity. But I'm not going to talk about any of that data because it's published, but it's been a kind of interesting wandering story using this model system C. elegans, which is incredibly powerful, both because it ages very quickly. As you'll see later, it's transparent, so you can do live imaging very easily in different age animals. But also now you can do phenomenal, well, you can always do phenomenal genetics, but with CRISPR now we can really start to do site-specific editing of endogenous genes in different tissues in different places and build up how these things work and ask causal questions about these genetic switches and aging. So this began as I was a postdoc looking, as was mentioned, at AMPK and trying to understand. This is a kinase which has many different targets. It's phosphorylated and identifies specific substrates of AMPK which are responsible for its effects on aging. We found one which is called CRTT1. There's 3 of these in you. There's 1 of these in a worm. It's a transcriptional coactivator. It binds to CREB, but other beta transcription factors too. And we found that if you block the effect of AMPK just from phosphorylation of two serines in CRTT1, that was enough to completely switch on and off the effect of AMPK on aging. Then when I started my lab, we've done a lot of work trying to understand where in the animal this is working. And quite surprisingly to us, a lot of that was really happening in, well, it depends if you think C. elegans have a brain, but in the neurons in the head of the animal, you can modulate these energy sensors just in kind of the brain of the animal. And that was enough to have a really dominant effect on the rest of the way the animal ages. So the perception of energy state and nutrient state in the brain was enough to regulate systemic aging in the animal across the rest of the tissues. We've been interested in trying to understand what receives those signals from the nervous system, and this is where we'll talk more about it today. And what we've shown is that really the energetic state in the brain then communicates to the mitochondria in different tissues, modulates their shape, modulates their function, and that is causal to how these animals are responding to these modulations in terms of their effect on lifespan. And I'll talk a lot about that. We've got very interested in the interaction between different organelles in the cell, how the mitochondria communicate both to the ER, to peroxisomes, a bit to lipid droplets, which I'll talk about today, and how that orchestration goes awry with age and can lead to metabolic homeostasis and defects in longevity, and how dietary restriction maintains it, which will be a lot of the focus of the unpublished part of this talk. And then some work which I maybe should have spoken to because of the institute here, but I'm not going to, on different sort of downstream homeostatic mechanisms like alternative splicing, RNA splicing, or in this case, kind of epigenetics, which Gio worked on in my lab and now I'm doing in his own lab, that are downstream of these kind of signals, these nutritional cues, which ultimately enable the animal to maintain its youthfulness. Anyway, that's all kind of published. Welcome to ask me questions about it. But now the rest of the talk is going to be unpublished and mainly work from one postdoc in my lab. It's really great. So I'm going to start here to the difference between these two. And some people love this, some people really hate it. If you hate it, you can tell me and put your hand up. But in the aging field a lot, we talk a lot about kind of loss of homeostasis with age, right? And the origin of the term homeostasis is by definition trying to keep an animal in this unique spot where everything's working perfectly, but it's a dynamic thing. The system moves out of whack one way and you bring it back in, and it can go both cases. But I think the term in the aging field anyway of homeostasis is kind of lost in translation slightly to kind of get to the state that there is more a static component that youth has that is lost with age. And if only we can restore this kind of youthful static place, this youthful place, that will help us to increase lifespan somehow. So like mTORC1 is in one state in a young animal, another state in an old, and we need to bring it back to this stasis. But really, that loses all the dynamism of the system. Biological systems are very dynamic. The way we're maintaining homeostasis is by dynamics of the system. And what I really think is lost with age is that dynamics, the ability of the animal or the cell to respond to different inputs, to have acute responses, and to bring the system back into balance. And so I kind of have begun to favor this idea of homeodynamics, and that what is really lost with age is the dynamism of the system at the cellular level, and that's maybe what we want to restore. And so I'll talk about what that means kind of conceptually and how it changes the sort of work we do. But if you think about something like mTORC1, right? There are lots of papers, including ours, where we can just genetically delete mTORC1 signaling. Give rapamycin to an animal, and that will just suppress mTORC1. But just suppressing mTORC1 is not mimicking youthfulness. That is a kinase whose job is to respond to multiple upstream cues, turn on and off in response to those cues, and do what it's supposed to do. So if you just render something static, that's not mimicking youthfulness. It's creating a different sort of dysfunction. That might have a positive effect on longevity, but certainly rapamycin treatment is not mimicking youthful mTORC1, right? That's just turning off that signal. So we've really begun to be interested from those kinases and how they respond, how they turn off and on in response to cues. With age, they become refractory to specific cues, and the dynamics of the system go with age, and how we might find interventions which modulate that. So when we think about these kinases, at their most kind of intrinsic job is to respond to an animal moving between feeding and fasting, to substrate input, right? So when we have fuel, one turns on, one turns off, changes the metabolism of the cell, the tissue, and then reverse it. So we're really beginning to think about the change in the dynamism of this system. So how an organism, for instance, responds to changes between feeding and fasting, which we do on a daily basis. That metabolic flexibility that you need to start burning different substrates, to go between burning carbohydrates and fat, and switching between those appropriately at different stages. What the mechanisms are with these kinases, I'll talk about organelle dynamics that respond to this metabolic flexibility, and how that might be lost with age, right? So that's what we really become interested in, not thinking there's one metabolic state that is youthful, but really that the flexibility of the system is the youthful thing, how that flexibility goes awry with time, and what we might do to restore that flexibility. Any questions? Yeah.
Where do you integrate reproduction in this whole paradigm? Because I view about energy metabolism from an evolutionary standpoint, the most critical aspect is to reproduce. And I remember kind of the fly experiments you showed. Those are virgins, like virgins in a mate, or
those. So the slide I showed, they were once mated or 24-hour mated female flies. It works for males too, but it works in sterile females too. So we did that. I did that in my PhD. So although there are huge reproductive shifts across dietary restriction, you go from reproducing a lot to not much. We don't think that simple Y allocation energy resource shuttling is causal to that phenotype. So you can at least get those sort of dynamic shifts in an animal which isn't investing a lot of energy in reproduction. Yeah. That doesn't mean reproduction is not, it's that kind of like the faucet versus the bath thing, right? It doesn't mean that the reproduction changes are not involved, but it's not an energy resource. A paper we published in C. elegans last year, I guess, for mTORC1 was using targeted protein degradation to degrade it in different tissues, and we could show that you could degrade it just in the brain of the animal, and then increase lifespan with none of the trade-offs to reproduction, none of the trade-offs to growth, none of the anabolic loss that you see when you do it everywhere. So I'm interested in that question, and I think certainly the evolutionary argument for why DR exists as a thing is that during famine, the chance of your offspring surviving is low, so you should shut down reproduction, upregulate stress resistance, and see out the hard time. Yeah. And none of these interventions would be fit from from an evolutionary standpoint, right? A diet restricted mouse is not a reproductively fit mouse. It's smaller, it can't maintain its temperature, it can't reproduce. But when we think about translating it to kind of human therapeutics, when we think about trying to do this in late stages in adults, maybe some of those trade-offs really aren't so important for the evolutionary fitness of that person. So that was a kind of very hand-waving question. But I don't think the simple Y allocation resource trade-off model that is so compelling doesn't have a ton of empirical. You can certainly, for some of the selection experiments in flies from the 80s and 90s, where you can drive selection in a lab and drive longevity, that all seemed to be shifts in reproduction. If you sterilize those animals, that was lost. For the more kind of direct targeting of nutrient energy sensors, it doesn't seem to be the case as much.
Just to clarify, when you do the treatment with rapamycin, these are for animals that are fed ad libitum, and you do see a full appearance like they are calorie restricted, or you do not?
So in the best rapamycin data come from mice, and in those cases you're doing rapamycin in mice that are given ad libitum food. They eat quite a lot of food. You get a massively robust lifespan phenotype in mice. It actually hasn't maxed out, so it mimics the extent of lifespan extension of dietary restriction. I don't believe that dietary restriction and rapamycin are interchangeable. If you look at the transcriptional profiles of those two interventions, they're slightly different. The trade-offs that come with both of them are slightly different than the caveats of them. We know very little, actually, ironically, about really how rapamycin is working to increase lifespan in any system, even if it's really through TORC1 what it's doing. But absolutely, those don't require dietary restriction. You can have, and there are lots of other ways to genetically modulate these components where you can get the effects with ad libitum calories going in. And in fact, even in the DR studies in mice and most things, it's pretty clear that it's not calories that are driving it, it's nutritional components and ratios of the components of food, so that's where it becomes kind of cool. Keep going. All right, so we were thinking about, moving between feeding and fasting states and the dynamics of the system. And one thing my lab has got very interested in, as I kind of alluded to, is mitochondrial morphology and how changes in mitochondrial morphology lead to changes in their function and to longevity. And it's both basic, this is this movement between fission and fusion, right? We've now known in the last 15, 20 years, there's been this huge kind of increase in the mitochondrial field, really getting beyond the textbook of these bean-shaped organelles that sit in isolation. They join forces, they break up, they interact with other organelles. And this movement between fusion and fission of mitochondria in different nutrient states or pathological states has a huge effect on what those mitochondria do, from ATP generation to interactions with older organelles and substrates they can develop. And your mitochondria in all of you also will move between different states at different times of day depending upon your fed or fasted state. And so what you need to have is a system that can keep this dynamic balance between these mitochondrial networks, that when you can acutely and appropriately move them to the right shape to change their function in response to the right input, right? And if you lose this balance through disease, through age, through hypernutrition, obesity, then you begin to see pathology. There's lots of examples of changes in mitochondrial dynamics, either hyperfused or hyperfragmented mitochondria that then become refractory to cues that lead to different kind of age-related conditions like the ones I put here. And so we really have become interested in trying to understand the contribution of the dynamism of mitochondrial networks to different aging paradigms, and really how maybe, and I'll mainly talk about this today, how that might be lost with age, how that dynamic regulation goes with age. And the reason C. elegans are a great model for this is they are this transparent organism which ages very quickly. So I see them as this kind of intermediary between in vitro cell culture studies where you can, or yeast, where you can do a lot of beautiful imaging, and maybe mouse studies where you can look at disease phenotypes. The C. elegans allow you to do live imaging in vivo in different tissues of different organelles in the same animal as it ages, as you give it a different food, different drug, whatever you want to do to it. And so we can create different mitochondrial tools. This is an example of one here. This is a muscle in C. elegans here, the body wall muscle, where we've tagged the matrix of the mitochondria with GFP targeted to the mitochondria, and then the outer membrane of the mitochondria with RFP. So you can begin to visualize these with fluorophores and look at what changes. We spent a long time building a ton of more tools now in C. elegans. This is all unpublished. You're welcome to have any of these tools. We're going to put this out soon, where you can use dyes to look at the network state of the mitochondria, ectopic expression of different things in different tissues, or now with CRISPR begin to tag different endogenous components of endogenous membrane proteins and look at mitochondrial state in the same animal in different tissues where they do completely different things as it ages and as you look at response. We have built a really huge toolkit now to kind of do cell biology in a living system that's aging, that's communicating with itself, that you can feed a diet, you can do different things. And so what we first want to do is just ask, well, what happens to those mitochondrial networks with time? These are pretty old data now from 2017. So again, looking at muscle, what you can see is that this is just a marker of the sort of fidelity of the muscle fibers in the worm. They don't really break down over this time period from day 1 through to day 9. But the mitochondria become very disorganized. They move from this interconnected network that progressively becomes kind of fragmented and then really disorganized. Either the machinery that drives fusion of the mitochondria or the machinery that drives fission of the mitochondria. So this is like mitofusins. There's 2 in us. There's 1 in C. elegans, SO1 in C. elegans, or DRP1, 1 in us, 1 in the worm, that drives fission. So you can delete these completely through genetic mutants, through DEG1 systems, and if you delete DRP1, the mitochondria move from being interconnected to really hyperfused. If you delete mitofusin, they move the other way to being very fragmented, right? So we can now use the genetics of the system to push the mitochondria wherever we want in a tissue-specific manner, in an acute manner with DEG1 systems of different type points. So then we can ask kind of causality about what do these changes we see with age, are they causally related to longevity phenotypes? And it's been a kind of interesting journey because we've seen that different longevity interventions require and utilize different mitochondrial network states for the longevity phenotypes that they induce, right? So this is what we talked about at the beginning, this activation of AMPK. This is in wild type animals that are eating as much food as they want. If you overexpress a constitutive active variant of this kinase, which really can't sense energy anymore, so it's always under a low energy state, it's mimicking the activity of that kinase that you see in a fasting condition. That's sufficient if you stick it everywhere in the somatic cells of this animal to increase lifespan. If you delete SNO1, which drives fission, or DRP, which drives fusion of the mitochondria, now AMPK doesn't increase lifespan on those animals at all. And so we published that work and we could work out why that was working. But if you take a different intervention, so in this case, inhibition of mTORC1, that increases lifespan. This is a genetic mutation of an amino acid sensor upstream of mTORC1. That's sufficient to increase lifespan even though they're eating a lot of food. Now, mTORC1 inhibition just requires fusion of the mitochondria. So if you delete the fusion factor, it doesn't work, but the fission doesn't matter. Delete the fission factor, it works just fine. So if you promote more fusion, mTORC1 still works. And the other case, if this is a lifespan modification, which is slightly counterintuitively deleting one component, one arm of the ERUPR, ATF6, that's sufficient to increase lifespan in C. elegans, but that requires fission. So there isn't a kind of youthful or a longevity mitochondrial network state, right? There isn't a good and a bad network, which is what people want to reduce things to all the time. It's a contextual requirement of different mitochondrial networks in different conditions, which enables some of these longevity interventions to work. And so that's all published, but what that really means is we can use, mitochondrial shape affects all these different things that mitochondria can do. They're not just, I hate the phrase powerhouse in the cell. Mitochondria do a ton of stuff. Many of them are put here in this figure, and just changing their shape, not even changing the mutational state of those mitochondria or the copy number, just changing the shape alone can affect all of those things. And now we can begin to use and are using these different interventions to ask, okay, if one needs fission, is that because it's particularly dependent upon mitophagy and damage, or if one needs fusion, is it particularly dependent on that? So we can begin to use these ways in this simple system to understand the phenotypic contributions of the shape of the mitochondria to those lifespan phenotypes. But I began this talk about homodynamics and the dynamism of the system, and telling you that mitochondria dynamically change through different states. And I'm getting a little bit bored of breaking things. So I'm a geneticist, and when you're a geneticist in a system like C. elegans, you want to ask causal questions. You do A, does it go through B, you turn off B, right? And then you ask if this is an epistatic pathway. And we've done that for a long time, but that's always breaking something. That's creating some really kind of synthetic state of the mitochondria state where you delete DRP1, you drive them one way to do epistasis. But it's not really getting at what happens in vivo in an animal as it ages, as those things are supposed to move, and really looking at the endogenous dynamism of the system and how that changes. So I'm beginning to get, I shouldn't say, a little bit over breaking things genetically, and want to look more like what's happening in an in vivo animal to these things. So what happens to the dynamics of mitochondrial dynamics? So what's interesting is that we can look at, this is again in a muscle, I don't know if you can see this very well, but we can move the animal between feeding and fasting. So just taking wild type animals with a mitochondrial marker, look at the mitochondria when you feed them, when you take food away, when you refeed them, And you can show that just as it does in vitro and cell culture, it does in mice, mitochondria move between different states. They move between fusion and fission as you move the animals. In this case, it's 48 hours of food, 48 hours of no food, and repeat. And you can see that you shift these mitochondria backwards and forwards. So we know they dynamically respond to changes in nutrient state. And actually, as you age them out, the kind of amplitude of that response becomes lower. So with age, it becomes a little bit refractory to those changes. But we can ask, well, what happens if we completely get rid of the dynamism of these things? And so what we did in this case is then delete both the fusion and the fission factors simultaneously. So now the mitochondria are interconnected. They look kind of like a youthful state, but they can't move. They can't respond to those changes. And ask, when we get rid of the dynamism, now what happens to the animal? So the first thing is the adynamic. So now if you move them between feeding and fasting, without this kind of, as you'd expect, they can't move, they can't fizz, they can't go to fission, they can't go to fusion, they don't move backwards and forwards. What's amazing is those mitochondria are not really broken. So you can look at the mitochondria. This is using one of the markers again in muscle. This is wild type animals when they're young. This is these animals where co-deleted fusion and fission factors. They still look kind of youthful. You can look at EM. They don't look particularly, look a bit weird by EM, but they don't look particularly disrupted. They still can consume oxygen. They can still respire. But what's amazing is now they don't respond from a longevity point of view to intermittent fasting. So intermittent fasting is just moving between food and no food, has been shown in many different organisms to be sufficient to increase lifespan. The dashed line here in black, that's intermittent fasting in C. elegans, increases lifespan, population alive versus age in days here by about 30%. By having adynamic mitochondrial networks that can't move, now IF doesn't work at all. So you need this dynamism of the system in a worm. We don't know where, but you know with what tissues we can do this now. But we need a dynamic response to the mitochondria in order for them to respond to these kind of dynamic interventions which increase lifespan, moving to feeding and fasting. And what's kind of wild is that's not true for chronic diet restriction. If you just give them less food to eat, but they can get access to that food all the time, adynamic mitochondrial animals respond to the ER just fine. So there's real functional differences between these shifting backwards and forwards type interventions that need dynamism and chronic DR. Yeah.
Can you tell me what do C. elegans die of?
If I had $1 for every time I was asked. No. I think it's hard to do pathology. They certainly, some of them die from kind of infection with time, age-related infection. I think there are certainly subpopulations of animals, in wild type animals, that die of different things. It's incredibly hard to really define cause of death in these things. We don't really know, and I think it's also, it's a problem. Fruit flies is a problem to really then ask, if you have a longevity intervention, is it shifting the same, it's the same distribution of causes happening, but at a later date was shifting different things. So it's a very unhelpful answer, but it's really hard to do actually.
So in this part, so this 30%, impressive 30%, the ones look better, I'm guessing.
So yeah, so that depends upon the intervention. You can try to have different parameters of health span in an animal. So it's locomotion changes with age, reproductive decline, different parameters of health you might imagine, even force if it's moving through a maze. I don't think we've done it a lot for IF, but for some interventions will just like stretch that period so they'll live longer and they'll be healthier to a time point, but they'll also spend a long period of time in kind of like a less healthy state. You just kind of stretch it. Some don't, some will just maintain healthiness but some die. So I don't know for IF that we've really fully characterized all those parameters of like the changes that happen with age. We probably should. I'm a little bit, I think there's conserved mechanisms that modulate aging across organisms. I probably think there are fewer conserved mechanisms that modulate kind of health span because the ecology of the animals is so different. So if I found a mechanism which maintained a worm in a real kind of like wormy, like a great worm, I don't know that it would work in a fly, but maybe that's just me trying to get out of doing.
With this experiment, would you argue that it's mainly affecting muscle?
No, I don't know that at all. What we're doing now with that, I think it's changing their capacity to burn different fuels in different states. And I think that really that's what they're unable to do. So when they move to fasting, they can't switch to burning fats very well. Where that's really working, I don't know if I think it's not in the body, it's in more metabolic tissues, so muscle, intestine. Yeah.
One question. So if I got it right, it seems that the double mutant is very important. I mean, for the mitochondrial dynamics, is very important for the lactation due to the temporary restriction. But why the double mutant doesn't impact the wild type?
Why questions are harder? So they're not dead. So you can have, right? I mean, again, it gets back to these are animals in a lab, in an incubator, given food, right? Clearly, they're less functional than mitochondrial dynamics is there for a reason. I think the reason is to be able to cope with kind of flux changes. But you can delete them, and in a good environment with no pathology, with as much food at constant temperature, they don't live shorter. This is even true in mice, actually. So there were studies in mouse. I took out so many slides, I'm going to still run out of time, but it's fine. Where people made like a mutant in mice that maybe had hyperfragmented mitochondria, and those animals will die really quickly in 3 or 4 weeks. And to rescue that, they kind of deleted the other side of the dynamics, and it would rescue that and age them. And they stopped the study at about 1 year, and the mice were still alive. So you can have mouse models where they're unable to move backwards and forwards, yet they're in a mouse house sitting in a hopper with loads of food, and they don't do that badly. It's kind of wild, right? But in this case, they're even a bit long-lived. The double mutant in some cases in C. elegans that is adynamic is a little bit long-lived, and we published a bit on that. We think that's kind of approximate effect like that.
Is the UPR activated in this?
The mitochondrial UPR?
Yeah.
My goodness, I should know that in the double mutant. But I don't, so I'm not going to answer it. I think we did it in the depths of that paper in 2017, looking at like HSP6, HSP60, and I don't want to guess. I don't want to give you the wrong answer, so I'm not sure. I'll find out for you. But certainly we haven't. My hunch is, and I don't think that these are mainly lifespan effects happening through induction of the mito UPR, but I don't want to answer it correctly, so I'm not going to. Okay, so then I'm going to move through now. Can't leave it last 15 minutes, but to get like, how do you get this loss of metabolic flexibility and dynamism with age? What really happens? And so this is a kind of RQ. If you put mice in a metabolic cage or humans, right, we can move between feeding and fasting and show that in a young animal they'll move between burning carbohydrate and burning lipids very quickly when you move between feeding and fasting. But in an old animal, that becomes more adynamic. You can't move between these different nutritional states. The ability to switch between different substrates is lost with age. And we can show that in mice too. If you put old mice and versus young mice, 4 months versus 23 months in metabolic cages, and you shift them between feeding and fasting and light and dark cycles, the kind of amplitude of that curve goes down with age. So your metabolic flexibility goes down with time. Your capacity to mobilize the right nutrients goes down with time. And we become interested in understanding, well, what's causal to that? What can't an old animal do in response to fasting that a young animal can do? And so what we did is we went back to the system that we like the most. We took C. elegans and we took them when they were young. This is young adult. It means it's kind of day 1 or day 8, which is like the equivalent of like me, kind of middle-aged. And we fed them, or we fasted them for a short period of time for 6 hours, and we asked what changes. How does the fasting response change in old animals? We did this first by RNA-seq. And what we were basically looking for, I'm going to show you the actual data, if I can get this to me forward, the kind of transcriptional response to fasting in a young animal and how that changes with time. And can we show maybe a blunting of some transcriptional response to fasting that might lead to a loss of metabolic flexibility with age? And all of this work now, so this one is done by Arpit, who's a postdoc in my lab. He'll be on the job market soon. He wants to stay in Boston, just saying. Has a K99, is really great, and he's done a lot of this work himself. All of this work himself. So the first thing he did is, these are now actual data, the last was just kind of hypothetical data, is just do kind of hierarchical clustering of the transcriptional response to fasting in young and old animals, and just to ask how they cluster. What you can see is that obviously you can really, first you can separate out in this unbiased way the young adults from the middle-aged day 8 animals. And in the young animals, you can really see which are fed and which are fast, but in the old ones, you begin to lose that resolution, right? And we showed with controls that the animals are still eating at that time point, many of the genes have changed, but you can't resolve as much the difference between a fed and a fasted animal in middle age. And so what ARPIT did is ask, well, what are the genes that are induced in fasting in young animals that aren't induced in fasting in old animals? What is the transcriptional response that we lose with old age, or C. elegans lose with old age? Thinking that we get lots of mitochondrial-related genes, and what he really found was a huge response in peroxisome-related genes, this other organelle which has been ignored for a long time, at least in the aging field until fairly recently, which is also involved in mobilizing lipids and burning very long-chain fatty acids, amongst other things. So what ARPIT saw, and this is some more data here, is that you take some clusters of genes which are massively upregulated in fasting but don't change in old animals, and many of these were components of membrane-bound proteins on the peroxisomes, right? So this is just some of these data in young animals, upregulation of these peroxisomal genes that you don't see in older animals. And so that has led us to become very interested in how peroxisomes might be a kind of early signal of a fasting response and causal to maintaining metabolic flexibility that maybe gets lost with age. And peroxisomes do a few different things, but one of them is around kind of ROS and redox, but one thing they do from a metabolic point of view is to oxidize these very long chain fatty acids before they can then be transported to the mitochondria and oxidized later. So we've become very interested in the last kind of year or two in understanding this maybe the role of peroxisomal fatty acid oxidation in the fasting response that might be planted in time. This is a key protein to remember, this PRX5, which is really responsible for all protein import into peroxisomes and C. elegans. I'll come back to it, but just know that PRX5 is massively upregulated when you fast a young animal, and it's not when you fast an old animal. So the first thing ARPIT did is just look at lipids. It's just take animals with a short-term fast when they're young and ask what happens to the lipids when you do this short-term fast. What he saw is you got a reduction in triglycerides and upregulation of diglycerides, but this proportion, the kind of those lipids that were changing were massively enriched for these very long-chain fatty acids that you burn in peroxisomes. So we're doing this now in old age and different things, but what that means, at least in a young animal that's fasted, that first response seems to be a real oxidation and shortening of these very long-chain fatty acids. We can then look at really, because transcriptional data is just transcriptional data, what happens to the proteins of the peroxisomes and what happens to peroxisomes themselves. So we flag tag endogenous PRX5, we can show with age that the levels of PRX5, this critical peroxisome protein import protein, goes down with time. It's just quantification of that on the right here. We then have lots of different in vivo reporters to look at peroxisomes by targeting GFP2 peroxisomes here, or using these sort of bimodal kind of reporters that get quenched or don't, depending on whether there's paxophagy going on. And what we can see is that peroxisome count goes down with age in C. elegans in the intestine. You mainly see peroxisomes in the intestine C. elegans. Also, if you have a fluorophore targeted peroxisomes, it becomes more cytoplasmic with age, suggesting that peroxisome protein import is going down, because you don't see these punctate peroxisomes with time. You get this increase in cytoplasmic signal. And then we want to understand, well, what happens? What's the consequence of loss of peroxisomal protein import with age on the metabolic state and on really, particularly lipid mobilization and nutrient utilization of lipids in a fasting state in those animals? So this is an intestinal cell in C. elegans where we have two different reporters. We have one that is regulated marking lipid droplets with GFP. Another one is reporting on peroxisomes with peroxisome targeted RFP. And you can see in young animals you get these punctate peroxisomes and these nice uniform lipid droplets. If you delete PRX5, if you degrade tag, I put it slightly out of order, but if you get rid of that protein, so if you synthetically get rid of that protein import, you get this kind of cytoplasmic diffuse red signal because that RFP can't go into the peroxisomes, and you get these very large lipid droplets. You get this like kind of obesity, almost like phenotype, where you get these enlarged lipid droplets. So we know that inducing peroxisome dysfunction in a way that mimics aging is enough to increase the size of the lipid droplets and create this kind of maybe lipid toxicity. We asked them what happens to the fasting response in animals that are wild type or have dysfunctional peroxisomal import. So this is now Nile red staining to just stain lipid droplets in wild type animals. You can see the lipid droplets here in the head proximal region of the intestine. When you fast them, those lipid droplets are burnt. They're reduced, the size goes away, the lipids are utilized for energy. So when you fast animals, just as true for us, as true for worms, you get this real kind of lipolysis effect of burning these lipids. If you don't have PRX5, if you have degradation of this through mutants or through degradation, you get both these very large lipid droplets that I showed you, but they're completely refractory to fasting. They can't mobilize those lipids, they can't use them in times of fasting, so they're just maintained. This is quantification like here. The wild type burn these lipid droplets very quickly, but when you have dysfunctional peroxisomes, that's not the case. So it really seems, at least in C. elegans, this key first step in mobilizing lipids in a fast is burning them at some point, at least involving the peroxisomes. And so this is a slide I should have put 3 slides ago. We want to be able to manipulate this system. So what we want to do now, because many of these things are critical for development, is get away from RNAi, get away from genetic mutants. We now do a lot of kind of targeted protein degradation in C. elegans, which is basically, this is the oxidant-inducible degradation system, where you take an endogenous protein, in this case PRX5, you CRISPR knock in a DEG1 tag to it, as long as it doesn't affect the function of the protein too much. You can then express a plant E3 ligase tier 1 in kind of whatever cell type you want. Still not much happens, but when you give this plant hormone auxin, it brings these two together and ubiquitinates your protein with a DEG1 tag on it and degrades it really quickly in about 30 minutes. And so these systems are really great because you can give auxin, degrade a protein, take it away, it comes back again, and just do it in one tissue and get control over the system. We've used this system for most of the TOR1 pathway, for mitochondrial fusion and fission, but in this case I'm just going to talk about PRX5. And so what became really interesting is that, one, we're interested in where those lipids go, right? So they're burned first the peroxisomes potentially and move to the mitochondria. So we wanted to ask, well, what happens to the mitochondria when you degrade PRX5 with this DEG1 system? So again, you can look at both the peroxisomes here with this RFP targeted peroxisomes and the mitochondria with the GFP tagged to the outside of the mitochondria. When you, either wild type animals, this is across many tissues in the worm, but this is muscle I'll focus on here, or when you degrade PRX5. Quite quickly you can see, without even quantifying this, which I'll do on the next slide, is that when you degrade peroxisomal protein import, the peroxisomes kind of functionally go away, but the mitochondria change really quickly. You get this complete swelling and change in the mitochondrial networks. So there's this interaction between these two organelles that's kind of really affecting both their functions. And again, remember, this is mimicking what we see in aging. We see loss of PRX5, we see degradation of protein import all the time. And so we asked, well, what happens in age with these things? We can show that in wild type animals, you get this progressive fragmentation of the mitochondria that I showed you right at the beginning of the talk. That happens across multiple tissues. But this progressive fragmentation of mitochondria happens much faster when you degrade peroxisomal protein import. If you do that in the background of PRX5 degradation, you get mitochondrial fission much quicker, and that goes much faster. This is quantification here. So that changing one organelle and mimicking what the age effects on peroxisomes directly and synthetically has a massive effect on this other organelle, which we think is so key to all of those different longevity pathways I showed you. So PRX5 degradation really drives mitochondrial fragmentation. And why I'm telling you this last couple of slides is because we're beginning to think that a lot of the causal effects we saw of mitochondrial fusion and fission on aging, lots of dynamics of that system, might actually be being initiated by changes to the peroxisomes happening first. Those initial kind of deregulation that we see in one organelle is maybe leading to the secondary thing. So these are data from a very old paper. This is now just doing DR by kind of chronic bacterial dilution in these animals. In wild type animals, dietary restriction increases lifespan. When you block mitochondrial fusion, so when you drive fragmentation of the mitochondria, dietary restriction doesn't work, and that's mimicking what happens to loss of peroxisomal function. So we wondered then whether these peroxisomes and the changes to this other organelle, which we haven't thought about so much, might be involved in this dietary restriction response. First of all, we could show that DR has a really strong effect on the peroxisomes. So if you see, wait, have I got this right way around? Maybe we'll screw up this slide. Let's look at time. So if you look at peroxisomes in ad libitum fed animals here, they're very punctate. By the time you get to ad libitum fed old animals, they've lost that kind of punctate staining here, and you've really lost peroxisome import. When you dietary restrict young animals, you have a youthful looking structure, and that's really maintained in old age. So dietary restriction maintains functional peroxisomes over time. It maintains peroxisomal import, and that will have a secondary effect on the mitochondria. So you can show that here. This is the age increase in kind of the cytoplasmic signal of this peroxisome thing. So when you see it goes up, peroxisome import is down. That goes up with time in wild type animals. It doesn't go up with time in dietary restriction. And then this is the last slide looking at, well, is this causal? Is this change to peroxisome protein import causal to dietary restriction? And it seems to be. This is wild type animals that are ad lib. Wild type animals are diet restricted. When we degrade PRX5, that's sufficient. This protein that imports the peroxisomes that completely blocks the DR response. So this is where we're kind of at with this unpublished data. I apologize, too much data, spoke too fast. But anyway, bear with me, you can ask me any questions. As we're building up this model of this kind of inter-organelle communication that's needed to mobilize lipids in a young animal. So in order to have a youthful fasting response, you need dynamic mitochondria, but you need dynamic peroxisomes, and the first thing that happens is this huge upregulation of protein import and potentially lipid utilization of peroxisomes, and this enables a fasting animal to take lipids from a lipid droplet, oxidize them through peroxisomes, and shuttle them to the mitochondria. With age, I think what we see is we see first an increase in peroxisomal dysfunction. That leads to these enlarged lipid droplets, which are refractory to fasting. You can't mobilize those lipids. It leads to deregulated mitochondria, and we're working through what that deregulation really is. And that itself really leads to an animal which is metabolically inflexible and now can't respond to many of these longevity paradigms that they are. So all these different phenotypes happen. So what we really think is about flexibility, right? So mitochondria can't just be static, sitting around doing nothing, consuming loads of sugar. What you really want is a flexible organelle that can do lots of stuff. I just want to play with AI basically yesterday. So you need like mitochondria made of like trees. I don't know what it wants, but you want like dynamic, flexible mitochondria with old age, and really it's that flexibility of the system which is really important. That's the end of my talk. This is my lab. A lot of this work was done by Arpit, who's sitting in here. Somewhere Gio's in here, so Arpit's there. Happy to take any questions. Yeah.
So you have showed that PRX addition has increased mitochondrial fragmentation much earlier, but when they looked at the lifespan curves, PRX addition still lived like one time.
Yeah. But even so even driving mitochondrial diffusion or fission doesn't really affect the lifespan of wild type animals. So when we have fragmented mitochondria, that doesn't lead to real short lifespan. In C. elegans, in fruit flies, it might be a bit different, but in C. elegans, it doesn't really change. What changes is their response interventions. Yeah.
Can you model exercising?
Different ways you can stick them in liquid and they'll start to just thrash, and you can do exercise. And people like Monica Driscoll have done like short-term exercise models, putting them in liquid for 30 minutes, 40 minutes, taking them out, and that increases lifespan, protects against Alzheimer's, models and C. elegans, all those sort of things.
Does that change the peroxisome?
I haven't done it. I haven't exercised much. There are lots of different ways to do it. It's super cool. I mean, it'd be fun to do. You're chairing, okay? I'll stop chairing.
Yeah.
Okay.
Yeah.
So this is a healthy cell. And what about the cells of very unhealthy, which we all eat, the cancer cells? So in terms of cancer cells, right, the ability of cancer to survive under nutrient stress condition. So, like how would the mitochondria adapt? Do you think the cancer cell actually has much more energetic and healthy mitochondria? But that's totally different from the conventional research. Cancer has a defective mitochondria.
So I very much want to get away from the kind of labeling of like healthy and defective mitochondria. There's a beautiful paper by Nika Daniel and Orion that's how you pronounce it, in EMBO, like a couple of years ago, right? And they looked at different cancer cell lines, and they have different metabolic requirements, right? So some of them are very oxidative, some are very glycolytic, different. And they have different requirements on mitochondrial morphology. So some of those cancer cell lines really needed fission, some needed fusion, and they really mapped out, and we're collaborating a little bit with Nika to look at this. So a lot of that is like the curvature of the membrane itself of the mitochondria affects how malach shuts down TBT1 and the lipids can be brought in, right? So different cancers have different dependencies, or this is just cancer cell lines, different dependencies on different mitochondrial states. So I think that's more how I see that kind of thing, is that it's not that mitochondria in cancer are deregulated or hyperregulated. It's just that in all cancers, they have to find a way to drive growth under conditions where they shouldn't usually, right? And some of them are going to be doing it by utilizing one substrate and others, and they'll hijack different mitochondrial pathways to get there.
So you're talking about the context specific.
I think so, yeah. Because I don't think there is one. I mean, apart from obvious things like Warburg effect. There's not one metabolic state in every cancer which is going to drive one mitochondrial state. Clearly, different tumor cell lines have different mitochondrial network requirements.
Speaking of breast cancer, we have a lot of lipid stored.
Yeah.
So you would think they will really be able to really utilize the lipid very well.
Yeah. But they wouldn't be able to. If there's lots of lipids, so the cancer has lots of lipid storage, or they utilize lipids for fuel in those. I don't know anything about breast cancer cell line metabolism. Yeah, it's cool. I don't know the answer to that.
Just one last quick question about.
Oh.
Hi, great talk. One question I couldn't see from here, but it looks like all the mitochondria, when you look at the muscle, the changes in the mitochondrial networks, are all affected by aging or by dietary changes? Because the muscle is characterized by different populations.
Yeah, no. So I didn't have any of that data. So no, and we're trying to do it. So this is. You want me to repeat the question, maybe? So it's whether mitochondria all kind of change in a uniform way with aging or the subpopulations. It's become super interesting in the field now about positionality in the cell of mitochondria that are tethered to different other organelles, have different functions, and how they will change. With these tools, we haven't done that. We have a paper going back soon using FLIM, like fluorescence of NADPH, endogenous fluorescence of NADPH, where we can visualize mitochondria by looking at fluorescence of that metabolite without any label at all. And we can use that in itself as an actual predictor of lifespan, but also we can look at, you can see subpopulations of mitochondria. We see heterogeneity in mitochondria coming up with age. So when you look at the redox state and NADPH levels in mitochondria, they're much more uniform with young animals, and they become more variant with age. I'm hesitant to say whether that variant is maladaptive or adaptive. I don't really know, but certainly specific pools of mitochondria are emerging more with old age, and I'd love to know. So really what we'd like to do is begin to, tether them to different organelles, really look at those sort of things. And you can do that in this system and try to ask that with age. I just think people should use C. elegans more for cell biology.
I would argue that from your model, you are missing the nucleus as an important organelle.
I mean, they've got, no, I completely agree.
And it would be fun to
yeah,
see
it
what
would be
the
amazing.
dosing
And so,
animals
yeah, these like perinuclear mitochondrial pools are super great. I went to contact site meetings to learn this, and I come away it's like, okay. But what's interesting about this kind of contact site field is still the focus, and this is true for the mitochondrial dynamics field, is really still on the mechanisms by which you have different contacts between organelles, the components of the different kind of bridges between organelles, much less on the kind of physiology of the organelle themselves, what those different mitochondria are really doing. And there, because you get stuck between the single-celled stuff and then the mouse stuff, and I think the one in the middle might be somewhere to go with that. So I'm super cool to do.
I think we can save the rest of the questions to directly after the talk. So thank you again a lot.